Thank you. So I must say that I'm presenting this paper on behalf of our PhD student, Martin. Martin could not be here because his visa was denied, unfortunately. But I just want to say that thanks to HFSP, we are supporting a team in Kenya in this project. And here is Martin. I'm glad to present his work. So what we are trying to do is to solve a puzzling biological question in a major disease. So we all understand what schistosomiasis is. It is a major neglected tropical disease affecting more than 200 million people worldwide, particularly in Africa. And it's a very complex disease in the sense that the life cycle itself goes through two intermediate hosts. The story begins when the cercaria released from the snails penetrate the skin. They travel to the liver where they mature, and the paired worms actually move down to the gastrointestinal tissues where the worms lay eggs, and hundreds of them every day. And one puzzling thing is the eggs penetrate the endothelium, the epithelium, and find their way into the gut. And they get released in the feces, and the life cycle continues. So this is a very complex process for an egg to accomplish, and we are trying to understand how it happens because an egg cannot move. So what we are doing right now, we have an hypothesis that we are trying to pursue this. We believe that the eggs actually recruit the host cells, and primarily the immune cells, the macrophages, fibroblasts, T cells. So we know they are immunogenic, so they will attract the immune cells to come to them and then piggyback them, of course. So we know the process of granuloma formation, but we believe there's a particular cell type, the fibroblasts, that play a critical role here, and that is what we are trying to digest. We think that the fibroblasts, due to the immune reaction, will become activated in some sense, and they will start to deposit the ECM and also create contractile forces that will drive the process forward. So this is the hypothesis we're trying to look into here, and we are doing this using both in vitro systems and in vivo systems. And this is the work that Martin is doing right now. He's trying to clarify this in vivo. And just to give you a glimpse of what he's doing, he has done a very tedious job taking mice at different stages of infection. So he starts from week 0 when he infects the mice, and then sacrifice mice time-wise through week 14 right now. So I will present to you just a glimpse of what he has done on this, and we are finding very exciting results. Of course, this is what you expect when you look at the liver of infected mice. You will see the granuloma forming, especially on the right side. You can see the sackled portions. Those are the granuloma. And we are trying to look around those granuloma and see what cell types are actually composing this and how they interact, especially the fibroblasts and the macrophages and the immune cells, and how they interact with the egg. So one thing that we have right now, which is exciting, we are looking at, you will see the cytokines, actually the major cytokines involved in immune regulation. You'll see them spike in week 6, and again we see another pattern in week 9. So we are trying to see whether this correlates with the migration of the egg in vivo. Thank you so much, and come to this poster for more discussions.